The last type of organic molecule we have to talk about are the nucleic acids. The nucleic acids are RNA and DNA. The main function of nucleic acids is information storage. Your RNA and DNA contain all the information you need to make the proteins in your body. So we just talked about proteins and how important they are and how you have to have the right amino acids strung together to make the functional protein. The information about which amino acids are in the protein and how to make the protein and when to make the protein and even how much protein to make, all of that is encoded into the DNA and RNA. It's in a secret code in the structure of the nucleic acids. The basic unit of nucleic acid is the nucleotide. So nucleic acids, whether it's RNA or DNA, are made of nucleotides. Each nucleotide has three basic parts. All nucleotides have a sugar, and they all have a phosphoryl or phosphate group, and they all have a nitrogen-containing base. So sugar, phosphate, and base are the three components that we need to have a nucleotide. We have 5 different nucleotides depending on which nitrogen-containing base we have. Sort of like we had 20 different amino acids based on what the R group was. With nucleotides, we have 5 different nucleotides based on the base. The 5 different bases that we can plug into the nucleotide are adenine, guanine, cytosine, thymine, and uracil. 4 of these are found in DNA. The adenine, guanine, cytosine, and thymine are found in DNA. 4 of them are found in RNA. We find adenine, guanine, cytosine, and uracil in RNA. So they both have A, G, C, but we have the T in the DNA and U in RNA. Just like we saw when we were trying to build a protein where we had to string amino acids together, if we're going to make a nucleic acid, we have to string nucleotides together. So we're going to join a lot of nucleotides together into a strand. To make a strand of nucleotides, we join the sugar of one nucleotide to the phosphate group of the next nucleotide. And as you can see on the screen, we have sort of a chain that goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and then those nitrogen-containing bases are pointing out to the side. This is one strand of nucleic acid. It has what we call a sugar-phosphate backbone holding it together, and then those bases are pointing off to the side. Just like we saw with the amino acid R groups that could interact with each other to make more complicated shapes in our proteins, these bases can interact with each other to form more complicated shapes in our nucleic acids. The bases can be held together by hydrogen bonds. The adenine or A bases can form hydrogen bonds with either thymine or uracil, so we have either A with T or A with U holding those together. And the cytosine forms hydrogen bonds with the guanine, so C and G always go together. In this figure, you can see some of the interactions holding 2 different strands of nucleotides together. You can see the hydrogen bonds between the adenine and thymine, and the hydrogen bonds between the guanine and cytosine. This is demonstrating the structure of DNA. So at this point, we're going to see some more difference between DNA and RNA. So I'm going to talk about DNA structure first, and then we'll talk a little bit about RNA structure. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. In DNA, the sugar is deoxyribose. So we can see the sugar in the DNA right here. That's deoxyribose. And the bases that are involved in DNA, we already said, are A, C, G, and T. DNA is formed by 2 strands of nucleic acids. So we have 2 different strands of nucleotides, and these 2 different strands are going to hydrogen bond together to give us the structure of DNA. We say that these 2 different strands of DNA are complementary. They're not exactly the same, they're not identical, but they complement each other, which means that if I have an A on one strand, there's going to have to be a T on the other strand for them to form hydrogen bonds with each other. And if there's a C on one strand, there's got to be a G on the other so they can form hydrogen bonds. So we have these two complementary strands of nucleic acid held together by hydrogen bonds between the bases. This is what gives us what we sometimes refer to as the DNA ladder, where the sugar phosphate backbones form the sides of the ladder, and then the rungs are formed by the bases held together by the hydrogen bonds. When this structure forms, it takes a little bit of a twist, and that's what we can see in the rotating picture. It forms a little bit of a twist, and that's why we call DNA a double helix, because we've got two strands of nucleic acid, and they're sort of twisted a little bit to give us the helix, the little spiral shape that we see. As I mentioned before, the function of the DNA is to hold the information about how to make the proteins in your body. And there is a ton of information encoded in your DNA. Every one of your cells has all of the DNA that's needed to make any of the proteins in your entire body. And it contains the information not only about the amino acids that have to be joined together to make the protein, but it also contains information that tells the cell when and how much of that protein to make, so that we can control it and make protein when we need it and don't make protein when we don't need it. The structure of RNA is a little bit different. So first off, RNA stands for ribonucleic acid, and that's because the sugar in RNA is ribose rather than deoxyribose. If we take a quick look back at this nucleotide, we can see the single change that makes the difference between ribose and deoxyribose. The sugar shown in the box on this slide is ribose, and you can see we have an oxygen right here. This is ribose. This is what we find in RNA. To make deoxyribose, all we have to do is remove this one oxygen, and then we have deoxyribose, what we find in the DNA. So RNA contains the sugar ribose. RNA is made of a single strand of nucleotides. We only have one strand of nucleotides instead of two. But this one strand of nucleotides that has all those bases sticking out to the side folds around and forms hydrogen bonds to itself, sort of like a polypeptide does, in order to form complicated three-dimensional shapes. So here on the screen we see a couple of examples of RNA structure. In this first structure, we can see that the strand of RNA starts right here where we see the one, and then it comes up this way and we can see all the A's and U's and G's, and it comes around here, and then it loops back on itself and starts forming hydrogen bonds to itself. So you can see how the further part of the RNA strand is hydrogen bonding back to the earlier part, and then it's going to form hydrogen bonds all the way around here, loop up around this way and form some more bonds, and we've got another loop held together by hydrogen bonds, and this is the end of the strand. So this is all one strand of nucleic acid that's folded back on itself and hydrogen bonded to itself to form the shape of that particular RNA. This side of the screen shows another example. We have the end of the strand here, and you can see this one's very complicated and folds back on itself in a number of different times and a lot of little loops and bubbles before we get to the end of the strand. These different shapes of RNA give us slightly different functions, but all of the functions are related to making proteins. RNA is short, temporary copies of sections of the DNA that we need to make a specific protein. So remember that DNA is going to contain all of the information you need to make all of the proteins in your body. The DNA is pretty special, so we keep it safely tucked away inside the nucleus of the cell so it doesn't get damaged. But it can't really do its job from in there. So instead we make copies of little sections of the DNA, and that's the RNA. The RNA is short, temporary copies of sections of the DNA that give us the information we need to make just one protein at a time. So we'll copy a little section of DNA into RNA. That RNA copy comes out into the main part of the cell where it can actually be used to help make protein. We have 3 different types of RNA with 3 different important functions in making proteins. The mRNA, or message RNA, is the section of RNA that actually contains the information about which amino acids are going to go into the protein. So it's got the instructions for which amino acids we need. The second type of RNA, the tRNA, which stands for transfer RNA, is a bit of RNA that actually goes out and binds to the amino acids in the cell and brings the amino acids back to the message RNA. So the message RNA tells you which amino acid you need, and the tRNA, or the transfer RNA, finds that amino acid and brings it to the message RNA. The third type of RNA is rRNA. That's ribosomal RNA. The rRNA is part of the actual ribosome, or the structure that builds the protein, and the rRNA is responsible for catalyzing the reaction that actually joins the nucleotides together. This is interesting because rRNA is one of the very few enzymes in the body that's not made of protein. Almost all the enzymes in your whole body are proteins, except inside your ribosomes you have rRNA that can catalyze a reaction, that can join two amino acids together when you're building a protein. Because our nucleic acids tell us how to build our proteins, and we already talked about how important it is that our proteins have the correct structure so that they function properly, we have to have the correct information in the nucleic acid if we're going to build the correct protein. When we talk about having a mutation, what a mutation is a change in the sequence of the bases of the RNA. So we change one or more of the bases in the DNA. Because the DNA tells us how to make protein, whenever we change the sequence of the DNA, we potentially change the protein. And whenever we change the protein, we could be affecting its function. So that's why we typically talk about mutations as being a bad thing. Because if you damage your DNA or mutate your DNA, then you could affect proteins, and when they don't work, things fall apart. But there are some mutations, typically much more rare, that can actually be beneficial. There are some mutations that could cause a change in the protein that actually helps it work a little bit better, or helps it respond to a specific situation more effectively. So some mutation can be a positive thing. Before we leave nucleic acids, there's one last molecule I want to talk about. And it's not actually a nucleic acid, it's actually a single nucleotide. But since we've talked about nucleotides in this section, I like to put it in here. The single molecule I want to talk about is called ATP, or adenosine triphosphate. We can tell that ATP is a nucleotide because it does have a nitrogen-containing base, it's actually the adenine base, and it has a sugar, here's its ribose sugar, and it has phosphate. But in ATP, adenosine triphosphate, we actually have 3 phosphates linked together. 1, 2, 3 phosphates connected to our nucleotide. This particular molecule is very high energy. It contains a lot of energy in its bonds. And the energy in ATP is used to run a lot of things that happen in the body. A lot of the reactions in your body that require energy, we get that energy from ATP. In order to release that energy so it can be used, we remove the last phosphate. So we start with adenosine triphosphate with 3 phosphates. When we sort of break off or transfer or move that last phosphate group, we're left with ADP, adenosine diphosphate. And that's a lower energy molecule. And the energy that was released we can use to do work in the cell. In order to recreate ATP when we need it, we have to take that ADP and hook that last phosphate back on to make ATP. And that's what we use the energy in glucose for. Most of the glucose that you take in is broken down, and the energy is used to make ATP, to join phosphate to ADP, so we get ATP, which again is the energy molecule that our cells use to do almost everything.